Okay, I will start now. Hello everyone, and welcome to the Medican Express webinar series. Today's topic is on chromatin factor research. We're going to focus on a group of chromosomal factors called the high mobility group A proteins in this talk. We're thrilled to have participants joining us from around the world. Whether you're tuning in from your labs or your at your homes, we hope you find these sessions helpful and engaging. And today we're very pleased to have Dr. Angel Bahadur Singh as our speakers. And Dr. Singh is the AMI Northers Research Group leaders of epigenetic machineries and the cancer research groups at the prestigious German cancer research centers. The center is also known as the DKFZ in Heidelberg. Additionally, he's the founding managing directors of Flaxo Aid Limited, a German consultant company for helping the big biopharma's R&D strategies. And Dr. Singh brings over 15 years of experience in the field of genomics. He's specializing in cancer epigenetics and immuno-oncology. And his extensive works has had a significant impact in the field of epigenetics mechanisms in development and diseases. And Dr. Singh's contributions to the fields, including the pioneering studies in the role of today's topic, the HMGA proteins in chromatin dynamics, and its research his research has applications in identifying therapeutic targets for cancer and fibrosis-related disorders. In today's sessions, Dr. Singh will present the important roles in this high mobility group A proteins in cellular differentiation, proliferation, and tumor progressions. Following the presentations, we will have the Q&A sessions where you can ask Dr. Singh's questions about today's topics. Please feel free to use the text chat functions throughout the presentations. When you can think any questions, you can just put the questions in the chat and we will see that. And before we begin, I would like to remind everyone that this webinar is being recorded and will be available for replays on social medias like YouTube. Okay, without further ado, please join me to welcoming Dr. Angel Singh for today's talk. Okay.
Thank you, Leon, for a nice introduction. So I just open my slides to talk about. Okay, this. Okay, what happened? Okay, so okay, now it's not working. So let me. Can you see the slide?
Yes, we can see the slides.
But
Yeah.
I'm not able to make it bigger. Okay.
I think it's the buttons on the lower corners, right-hand side corners. This
Actually, no, So wait a minute. Before it was working perfectly, and now I see this problem that I'm not able to make it bigger from here. Okay. Let me find another way.
Yeah. Okay. Yes.
Now can you see it?
Yes, I can see it clearly.
Yeah, that's great. Okay, thank you, Lian, for the nice introduction. As you know that today I'm going to give a talk on high mobility group proteins and what we learned the lesson from that. Just before proceeding to that, I just put it here a general disclaimer, being that though I'm an academician working at German Cancer Research Center, Heidelberg, but I also founded the company, so it's just a disclaimer for that. So just to make it clear that what you are going to learn today. So today, basically, I'm going to talk about high mobility group A proteins and its molecular network in development and disease, and the function of HMGA proteins and its molecular network in gene transcription, as well as I will also highlight some of the future directions in the chromatin factor studies that I'm following. So as you see in the right side, there's a two word came up, HMGA, and it's a negative upstream regulator, billet saver. So most of the topic today will be around this. So with this, I would like to first introduce to make it clear that what is chromatin factors, because it's really important to know. And chromatin factors are basically kind of another kind of proteins, but they are present in the nucleus. They work alone or in the form of complex, and they interact and modify chromatin. And there are different types of, for instance, there are architectural transcription factors. They influence chromatin compaction and structure. DNA binding factors, they bind to DNA to influence chromatin structure. Histone variants, they are specialized histones for specific genomic regions. Histone chaperones that facilitate histone deposition and nucleosome assembly. And chromatin remodeling complexes, those are involved in to reorganize nucleosomes for DNA access. Histone modifying enzymes, those modify histones to alter chromatin structure. Having the sense there are different type of these chromatin factors, then the question comes to our mind, why someone should research these chromatin factors. So the first of all, these chromatin factors, they are able by regulating gene expression to control cell identity and function. They also influence cellular memory and behavior. These factors also, they get dysregulated in specific disease condition, and to understand them, one can also understand disease mechanisms and also developmental processes. And these factors one can also target, and they could be new potential treatments in diseases like cancer or fibrosis. Now we'll give a small introduction about high mobility group A proteins. So basically, high mobility group A, this is a family that is comprised of two family members, HMGA1 and HMGA2. They are basically non-histone chromatin architectural factors. These proteins has 3 80 hook DNA binding domains. As you can see in the right side, they are spaced as 80 hooks, and these basically DNA binding domains, 80 hooks, they binds to minor groups of DNA or 80 rich regions in the genome. As also it has acidic domain as a C-terminal tail, and that also influences affinity to binding to specific regions, as well as it also has impact on interacting partners whom to interact with. And they basically have diverse function. For instance, this regulates gene expression, they can alter chromatin structure, as well as these proteins also exhibits DRP AP site cleavage activities. Basically, upuric sites are generated during DNA damage repair. And this protein has this lyase activity, with that also it can cleave the DNA. So the most important thing to understand about this protein, that these protein are developmentally regulated. What does it mean? They express in the early stage, but in the somatic cells, they either express at a very low level or they don't express at all. And when it comes with a biological significance, they involve in development, tumor formation, diabetes, and obesity. And the question comes why I decided to work on this. So basically, we had already all this information when I started working on this protein, but still, there's a lot of things still not known, and one can ask one specific question to begin their journey in the field of science. And this is basically what happens with me almost 15 years before when I started my PhD, basically. And my supervisor basically gave me one question, and with that, my research journey began. And the question was, what is the function of HMGA2 in lung development? For that, basically, we are using murine model. And as a matter of fact, if somebody asks what is the function of that specific protein under a specific condition, then one should ask themselves that, do this protein at all present in that given time frame or to that specific cell or to which specific cell type? And for that, basically, I did the very first experiment to see where HMGA2 is present into the lung during developmental stages. As you see in the figure that E12.5, that's embryonic day in mouse, the expression of HMGA2 is quite ubiquitous, but E14.5, it's to the specific region, and that's more to the distal airways that I'll express, and that we further confirmed at protein level by immunohistochemistry that indeed HMGA2 present, because just knowing protein present, gene present at RNA level, given the fact that the function of this gene is by expressing protein, then one should also check at the protein level. So once we confirm that indeed HMGA2 is present in the mouse embryonic lung at the distal ridge, then we ask ourselves, so what they are doing there? And we did several experiments and we found out that basically we using like here we are presenting the data with HMGA to knockout mice, global knockout mice. And there we observed that there is an increased level of canonical, increased level of beta-catenin in the nucleus, one of the hallmark of showing the highly active canonical wind signaling, as well as we used bad-gal transgenic mice. These mice are reporter strain that express beta-galactosidase in the presence of activated beta-catenin. And we saw that in the HMG2 knockout mice lung, there's a hyperactivity of canonical wind signaling. And with additional data, we confirm that basically HMG2 fine-tunes this canonical wind signaling during lung development. And then we ask ourselves how. Means we see that is expression, and then we see that it has to do something with fine-tuning of wind signaling. Then we ask how, and we found out that basically HMGA2 directly regulates GATA6 gene expression. And this GATA6 gene basically directly regulates Frizzle receptor D2, FZD2, that was published few years back to our research by this Yang et al. In Nature Genetics. They showed that basically GATA6 fine-tunes wind signaling if in epithelium by regulating Frizzle receptor FZD2. And we found one layer ahead upstream that basically HMGA2 directly regulates by binding to the promoter of GATA6. And thereby basically it involved in the fine-tuning of chemical wind signaling in lung development. Being a curious scientist, I ask next question: how does HMGA2 participate in GATA16 transcriptional activation? And I was not much aware about where to begin to address this question, and I decided to proceed with unbiased HMGA2 interactome analysis using mass spectrometry. And in this analysis, I identified several interacting partners of HMGA2, including ATM and H2X, that we also validated by immunoprecipitation assay, as you see in the right side, that indeed HMGA2 interacts with ATM and H2X, as well as its phosphorylated form of H2X at serine 139. Basically, H2X serine 139 phosphorylated form is, in general, we call it DNA damage marker gamma H2X. And we got an opportunity there to investigate the effect of HMGA2 together with ATM and S2X and its phosphorylated form of S2X, gamma S2X in gene transcription. And when we begin this study, there was no report indicating that gamma S2X plays even a role in gene regulation. So it was but obvious for us then to really see that if HMGA2 recruits ATM and phosphorylated form of S2X at the promoter of GATA6. And for that, we perform chromatin immunoprecipitation assay. And as you can see that basically HMGA2 loss has an impact on the enrichment of phosphorylated form of S2X at GATA6 promoter, as well as it regulates also the ATM enrichment at GATA6 promoter. We also perform mutation analysis where we basically mutated phosphorylated form of S2X to hypomutant by changing serine to alanine, so it cannot be phosphorylated. And we saw that when we mutated S2X as a mutant, and we saw that HMGA2 was not able to increase the expression of GATA6, indicating that indeed HMGA2 mediated or S2X phosphorylation required for HMGA2 mediated GATA6 transcription. One thing is to knowing whether the effect what we see is really one specific gene or it's a general mechanism. And to show that the finding which we made specific to GATA6 promoter and its activation by HMGA2, ATM, and phospho-S2X involvement. We also saw by the chip sequencing and global analysis that it's a general mechanism. As you can see that there's a promoter region, which is we see the enrichment of phospho-S2X is reduced in the HMG2 knockout from 7.5 to 4.7%. As well, we saw that knockout has impact on several genes. Those are being regulated by HMGA2, indicating that indeed it's a general mechanism. With this, we come up with our first mechanism, where we saw that HMGA2 basically interacts with ATM, and this interaction helps in the phosphorylation of S2X. And this phosphorylation is essential for transcriptional activation of HMGA2 specific target genes. And this work we published in Cell Research in 2015. And we further analyzed basically our ChIP-seq data. As you can see that very nicely, how HMGA2 loss has an effect on the enrichment of phospho-S2X only specific to the promoter site. And this also gave us the next question to ask, basically, does HMGA2 also participate in S2X deposition at this target gene promoter? And for that, basically, we look back to our mass spectrometry-based in HMGA2 interactome data set, and we identified that HMGA2 also interacts with fact complex component SUPT16 and SSRP1. Basically, it's a well known that fact complex involved in S2X deposition into chromatin. Then we ask whether HMGA2 loss has also impact on enrichment of these components on the promoter of GATA6 as well as other identified targets, for instance, mTOR, IGF-1. Here we use our Raptor as a negative control to show that the non-target of HMGA2 has no effect on the enrichment of this complex component. And this we showed by the chip data, and with this we concluded that yes, indeed, basically HMGA2 complex interacts with the FACT, also involving into enrichment of this FACT complex to their promoter for S2X deposition to the promoter, and then ATM basically comes to play a role where ATM phosphorylate S2X and is essential for transcriptional activation. So just to summarize what I talked so far, so what I talked that HMGA2 interacts with FACT, ATM, and with that basically involving S2X deposition and phosphorylation, and this is essential for GATA6 gene transcription as well as other downstream target of HMGA2. Now, basically, I will talk about MiCEE, which is negative regulator of HMGA2. And this we started investigating at the same time, and we were hoping different mechanism, but you will see that how what we study sometimes make us to follow another path, and then understanding how one can combine both to reach to one common goal. So just for the curiosity, we looked the expression of MiCEE by immunohistochemistry, by FIS, microRNA FIS, as well as by nuclear and cytosolic fractionation. And we were surprised to see that basically MiCEE is present also in the nucleus. And then we ask ourselves very first question that anyone asks, if something is present somewhere where you don't expect, because normally the microRNA plays a major role into the cytoplasm by regulating the degradation of mRNA in cytoplasm by RIS complex and mediated by AGO2. And we found that this MiCEE present into the nucleus. And then we ask ourselves the question, what is the function of mature MiCEE in the nucleus? And to address that, we had no clue which direction to follow. And then we ask ourselves, so what strategy we could do? So we did two things. The first thing, we performed RNA sequencing after microRNA 70 pull down, as well as we did nuclear RNA sequencing. And with that, we identified microRNA interacting nuclear RNAs, as well as microRNA less 70 regulated nuclear RNA. So the figure B and C talks about the data what we got from the microRNA pull down. As you see that around 7,500 in total RNAs we detected, and out of that 82% those was interacting with MiCEE was coming from non-coding RNAs. And when we further analyzed, we saw that around 38% of these non-coding RNAs was coming from some promoters adjacent to coding RNAs. And in that we saw Around 19% were bidirectionally expressed non-coding RNA and coding RNAs, and 19% was monodirectionally. And we decided further to investigate bidirectional expressing non-coding RNA and coding RNA because it was easy to design the experiment and to follow the next question. And what we observed that basically microRNA 7D primarily binds to non-coding RNAs expressing to these promoters. However, at the expression level, we see the effect on both non-coding RNAs as well as coding RNA. Both were reducing in terms of their transcriptional expression. Then we ask ourselves why we do see that and what could be the mechanism. So for that, we proceed with mid-7D interactome analysis by mass spectrometry, where we pull down protein, those were interacting with microRNA mid-7D, and we analyzed and we identified that majority of the genes, those were coming, proteins, those were coming as interacting partner of mid-7D was from RNA processing, RNA splicing, RNA localization, chromatin organization. And in the RNA processing, we saw that majority were coming from the exos complex, basically exos complex or DNA degrading complex involved in RNA surveillance. And we also nicely see that mature microRNA by FISH combining with immunohistochemistry, expressing, co-localizing into the nucleus with exostain. One of the nucleus is specific factor of exos complex. And this actually gave us an idea that most likely mid-7D degrades non-coding RNAs as well as coding RNAs into the nucleus. And to address that, we performed again a small RNA sequencing into the nucleus to identify the degradation product of RNA, which potentially mediated by mid-7D. And it was really surprising for us to see that basically mature mid-7D reduces only the level of non-coding RNAs into the nucleus by RNA exome-mediated degradation. However, we did not see the effect on the coding RNAs. And this led us to ask, okay, so what we see the effect on the non-coding RNAs is not the same for the coding RNAs. So it means that most likely mid-7D mediated non-coding RNA degradation triggers transcriptional repression. And most likely because of that, we see the effect on the reduction in the coding RNAs, which is independent of RNA exosome-mediated degradation. With this hypothesis, we further investigated. We looked to again interactome data set of mid-7D, and we saw that it's also interacting with the PRC2 enzyme component, enhancer of geste homolog 2, EZH2, and we saw that basically the downstream target of mid-7D basically also when we overexpress mid-7D, we saw that enhancer of geste was also enriched to the promoter of downstream targets as well as its histone marks S3K27 trimethylation. And when we make a loss of enhancer of geste homolog 2, we saw the opposite effect, confirming that most likely mature mid-7D reduces non-coding RNA level in the nucleus by PRC2 mediated transcriptional silencing. And with this, basically, we came up with some idea that there is a mid-7D, there is a exos complex, and there is a PRC2 complex. But they somehow work together, and then we ask whether they form a complex to execute their function. And for that, the first we ask ourselves, as a MID-7D is a microRNA sequence, it has a seed sequence, whether it binds to the non-coding RNAs by complementarity. And for that, we perform the mutation analysis, and we showed that indeed, basically mature MID-7D binds to non-coding RNAs by complementarity to form MID-7D non-coding RNA duplex. And we also identified that there is a cofactor of Exos complex C1d that basically binds to MID-7D non-coding RNA duplexes, and that we confirm by IMSA, where we perform the competition analysis, as you can see here. And we also saw that when, as you see in the figure D here, when we added Exos 10, that this degrades the bound probes where you see the C1D and MID-7D, for instance, here. And when we added the quencher EDTA, which is basically quenches the divalent cations magnesium, which is essential for exhaustion activity, and we saw that it came back indicating that yes, C1D basically binds with MID-7D non-coding RNA duplexes, and these then associate with exhaustion, because it was already known that exhaustion interacts with C1D, and then it exerts its degrading activity. And further, we showed that basically exhaustion works as a interactor for C1D, which basically interacts with MID-7D and non-coding RNA duplex, and exhaustion also interacts with enhancer of sustain. As you see here, we performed mutation analysis as well as sucrose-based gradient assay to show that all the components are present together, as you see in the figure C. And then we perform mutation analysis, and we saw that exhaustion basically interacts at the N-terminal domain with C1D and to the C-terminal domain with enhancers of sustain homolog to BJS2. And with this, we coined the one term we gave to this complex, which comprised of microRNA MID-7 exos complex and PRC2 complex with distinct functionality. That microRNA MID-7 basically provides genome-wide loci specificity to target this whole complex to a specific genomic loci. Exos complex comprising of other components along with exhaustion and C1D, the core main component of into the nuclear compartment involving the non-coding RNA degradation and PRC to mediate a transcriptional suppression of non-coding RNA by heterochromatin formation. And this is just a summary of what our finding is. Once again, just to explain that microRNA basically binds to these bidirectionally transcribed long non-coding RNAs. These duplex then bounds by C1D, which further recruit exos complex enhancer of sustain for the repression of downstream target genes into the nucleus. Not only that, we deleted these non-coding RNAs, and we were surprised to see that basically not only this complex involving transcriptional suppression, but it also involving tethering these silence genes to perinuclear heterochromatin domains around the nucleolus. Sorry. So with this, basically, we just found a new mechanism of microRNA working in a complex of MiCEE into the nucleus. However, we were not so sure about its functionality, that whether it only has a transcriptional mechanism or it also has some biological relevance, either in context of cancer or fibrosis. And here I'm going to talk now about the model which used lung fibrosis to test this, the new complex that we found. Just a brief information about the lung fibrosis is a progressive and chronic lung disease characterized by thickening and scarring of lung tissues. The cause of lung fibrosis is not much known, but it includes environmental exposure such as occupational dust, autoimmune disorders, medication, side effects, and certain infections. And there is no cure for lung fibrosis. Various treatments only aim to manage symptoms and survival time of 3 to 5 years from the time of diagnosis. So we ask whether MiCEE complex has a potential as a therapeutic target or not. So the first thing for us to know if this complex is present into the healthy lung as well as in the fibrotic lung. And for that, we used a very simple analysis. We had idea of about downstream target of this complex, and we simply look into the RNA-seq database of human lung from the fibrotic and the healthy patients, and we saw that majority of MiCEE complex downstream targets were upregulated in human lung fibrosis, which gave us a very first clue that most likely MiCEE complex is inactive or disrupted, but we had to experimentally test that. So what we did, we used the human normal lung fibroblast and IPF isolated. IPF here is idiopathic pulmonary fibrosis, lung fibroblast, and we did sucrose gradient based fractionation. We looked for the enrichment of component of MiCEE complex, exhaustion enhancer of gestation C1D, as well as meatless 7D. As you can see in the left side, they are nicely enriched and they are coming to one fraction. In addition, we also found that this complex also somehow interacts with HTEC1 and HTEC2 coming to the same fraction. And also it exerts as a activity that give us further easy to analyze further effect here. And when we saw into the IPF, we saw that this complex or somehow dismantle, as well as we saw that marker RNA mutation also low expressing into the fibrotic samples, as well as we saw that there was a less STAC activity, indicating that this complex is disassembled in idiopathic lung fibrosis patient. Then we ask us the next question, what could be the mechanism of this disassembly? The one thing we knew that microRNA midless 7D is reduced in the lung fibrosis. So we ask, what if we add this component, can we reconstitute back our complex there? And yes, we saw that was indeed the case, that when we put midless 7D back, it regained to a certain extent the MiCEE complex. The next, we ask what could be most likely the potential additional reason going away from the midless 7D reconstitutions. And in this process, we found that basically into the lung fibrosis state, there is excessive presence of hyperactive EP300. And we knew that from literature that basically EP300 involved in modulating the activity of STAC, as well as it regulates the activity of Lin28, which is basically the regulator of microRNA. So that gave us this candidate as a top candidate to further analyze. And what we simply confirm the first excessive presence, as you see in the immunohistochemistry analysis, the control, you see the EP300 and activated P300 is much more excessive into the fibrotic IPF, ACTA, ACTA2. This is basically the marker, alpha-smooth muscle-actin marker for the myofibroblast or hyperactive fibroblast in IPF. And indeed, we saw that there is a excessive presence of hyperactive P300. And then We also confirm that if we block EP300 by specific inhibitor, can be increased as the activity, and it was indeed the case, as you can see here, that when we block with P300, we saw that uh, there was a increased activity of EP300. Here is just for you the control placebo group and IPF placebo. You see that there's a less activity, though the left side, if you see, stack level, stack 1 and 2 level was mainly that stack 1 was very high in IPF patients. But when we look to the activity of this stack 1, it was very low, mainly in the nucleus, indicating that what we see at the protein level, it does not usually reflect what happens with in terms of its enzymatic activity. If this specific protein contains certain type of enzyme activity, and that was very clear for us here, that though there's a high level of stack into IPF, but activity was quite low into the nucleus. And we were able to reconstitute back when we inhibited with EP300 inhibitor. And then we ask ourselves, can we, just by inhibiting EP300, revert lung fibrosis to healthy in vivo condition? And for that, we used bleomycin induced murine model, lung fibrosis murine model, where we basically treated with EP300 inhibitor. And as you can see here, basically that you by the serious rate and collagen content analysis, we nicely saw that basically treatment with EP300 inhibition reverts lung fibrosis to healthy in vivo condition. With this, I would like to summarize this part of the talk where we saw that in healthy state, MiCEE complex is intact and functional. In lung fibrosis, due to hyperactive EP300, inactive stacks, and low level of MID-7D, this complex is non-functional and disassembled. Nevertheless, one can reassemble this complex back either by inhibiting P300 or by the gain of MID-7D, and thereby one can reverse the lung fibrosis towards healthy state. So just to summarize so far what I talked here first about HMGA protein, its role in the lung development, and then I talk about how it's directly regulating GATA6 by recruiting ATM FACT and by phosphorylating S2X, showing it's not a gene-specific mechanism, but it's general mechanism, and afterward discussing about what we found completely different paradigm with its one of the negative regulator, MID-7. So if someone asks about what could be the future direction based on these findings and what we are basically pursuing, also I'm looking collaborators basically who could join my hands or our hands, let's put it like that, to follow a specific one of these future direction. To summarize, I put here only 3 directions, but there are more. The first one, to just ask about what we found out in terms of mechanistic finding, that HMGA2, FACT, ATM, and gamma S2X axis in gene regulation. And we had observation that all these components of these axes are highly expressed in subset of cancers and correlate with poor overall survival. One can see into the CBAR portal. This is publicly available where anyone can check. And this gives the open question whether this axis drives cancer and its impact on tumor microenvironment. And can this axis be exploited for cancer therapy? It's a still open question. Part of that I'm pursuing in a follow-up project. However, I did not show this here because the data is still not published. And the second one is mechanistic finding that we had, that HMGA2 directly regulates GATA6 during embryonic development. My colleague, when I was working, also in that also I'm a co-author. They has shown very nicely that embryonic isoform of GATA6 is silent in adult lungs, but reactivates in lung cancer, making it a potential biomarker. Published in 2016 and 2023 as a first author by my colleague Aditi Mehta and Rubio Carla. And that gives an open question that we did not follow so far, that whether HMGA2 regulates this GATA6 isoform switch, and whether this isoform switch is a general mechanism in cancer. If it's general mechanism, can this be adapted to develop potential biomarkers, not only in lung context of lung cancer, but also in context of other cancer like pancreas, where HMGA2 is highly expressed. The third mechanistic finding that I would like to talk about, our finding that hyperactive P300, which causes MiCEE complex disassembly in lung fibroblast. And we have observation that fibrotic fibroblasts are excessively also present in the tumor microenvironment. And it keeps the open question whether this mechanism drives tumor growth, and can EP300 MiCEE axis also be exploited for the cancer therapy. Some of the parts basically in these directions I'm following in my lab, and some are still not pursued. And I would be happy to collaborate with some interesting future researchers who would like to follow one of these directions. With this, I come to my acknowledgement part, where I would like to express my gratitude to Professor Guillermo Barreto. Basically, I started my work there, and then I moved there at Max Planck Institute for Heart and Lung Research, and then afterward, I moved to German Cancer Research Center, DKFZ, in the department of Professor Hackenwalder, where I'm also pursuing part of this project, as well as I would like to express my acknowledgement to my collaborators who basically involved in success of this story building of this research. With this, I would like to say thank you. And I'm open for the question.
Okay. Thanks for Dr. Singh. Yes, we have one question here in the chat from Sergei Glokov. Thank you for the interesting presentation. The question is, the S139P H2AX is a product of ATM activations. In the case of a double strand of DNA breaks repaired. The ATM is the main DNA repair protein kinases. Is there any link between the high mobility GA2 activities during the embryo developments and the DNA damage controls and repair signaling pathway, you think? Including the chromatin modeling and the DNA repair factors, the orchestration of recombinant partners joining together. Do you think this the HMG2 have a role in DNA damaging pathways?
So, if you ask me about this question, so what I can tell you that during transcriptional activation, mainly those are happening by active DNA demethylation process. There is a massive involvement of DNA repair mechanism. For instance, in 2008, there was a paper by Guillermo et al. In Nature that showed that GAT45 basically involved in the active DNA damage. So they exploit a DNA damage repair mechanism for active DNA demethylation process, and which is very essential for transcriptional regulation. So in this aspect, when I say that during transcriptional regulation, that's a very specific and very unique situation where we see the effect of HMGA2 ATM mediated gamma S2X involvement for transcriptional activation. However, when we talk about the DNA damage as a whole, so basically the ATM is also the kind of a guardian for regulating the DNA damage control, and most likely they do have the role also in the DNA damage repair as a specific context where the due to toxin exposure or due to replication happening some DNA damage. This I cannot discard. There is also the possibility. There was also a paper in 2015. There also a group has shown that basically gamma S2X also linked with the neuronal activation. So there's also another group evidence indicating that S2X phosphorylation involvement with the transcriptional activation. So I would not say that this is happening this way or that way. We basically comes from the gene regulation background, and we did our observation the very first time that what is involvement of ATM and gamma S2X in a transcriptional regulation. Though these proteins are well known into DNA damage community that they are involved in DNA damage repair. So this we cannot discard. And all these mechanisms basically happens hand in hand in development, in disease condition, as well as during the homeostasis into the adult cells. And one have to really look with a different specific, depending on the question what you ask, you get the answer in that specific line. And for us, it was really interesting to see their involvement in the transcriptional activation, and that was very new for us. So we decided to follow this direction.
Thank you. Yes, I think Sergey, he has another questions. Is there any relationship between the HMGA2 ATMs and then the YH2AX complex activities during the development of organs? The ATM and YH2AX cooperation during the spontaneous DNA damage repaired for one and the same embryo. I'm not sure I quite understand the question. It's in the chat. You can look.
Yeah, actually, I'm also not able to follow this the question, but what I can tell that most of the studies has been done independent, like either looking for the function of HMGA2 or just ATM or just S2X. However, there are not or very few studies has been done where people try to combine. And of course, this is a very open question still. As of my knowledge, I don't know if someone really did this trio axis, HMGA2, ATM, and gamma S2X axis activity analysis in development by combining knockout model of HMGA2, ATM, and mutant of S2X. So as of my knowledge, I don't know if someone has done it in this direction or not. It's still a very open question. Because just here I wanted to add that sometime we use, people talk to use about organoid models or this model or that. So each model, it has their own drawbacks. But real understanding, if someone really wants to do in the developmental context, then one should really follow the live organism like either the murine model or the zebrafish model, where they can, or the Drosophila model, where they can really see all these components playing a role together. And in my knowledge, no one have done any analysis in this direction, combining these axes.
Okay. Dr. Singh, actually, I have a question. I have two questions from my side. The first, in your the IPF models, it shows that the disassemble of the MiCEE complex is because of the three factors: the hypoactivities of P300s, and then the reductions of the MiR-led7D, and another one is the acylations. So do you think there's like a more intricate relationship between maybe it's the P300 cause that reduction of the somehow the MiR-led7D, or do they like happen in different through different pathways, and then both three conditions have to happen before this start to disassemble?
Actually, we did not address this question so far, but what I can tell you based on the information what is present in the literature, that yes, most likely ATM, no, sorry, this hyperactive P300 negatively regulates the level of mid-7. Now the question comes to the cytoplasm or to the nucleus. We did not follow this direction. We can't say this. And what we know that this P300 also involved in the regulation of Lin28B. That's also one of the essential protein involved in the bioprocessing of microRNA during maturation from precursor microRNA to the mature microRNA. So most likely it's coming also directly affecting the level of mid-7D. And it could be also a completely independent path they are following. And we did not dig in detail about this upstream regulation of mid-7D, how the link between mid-7 and P300 is in the fibrotic condition.
Okay. Yeah, I have one more question, the technical questions, because I
Yeah.
saw you from your systematic study for the RNA interactions. I saw you could detect many thousands of interaction between the target RNAs. I'm just wondering if that's in a context that's because I don't know the detailed procedure. Do you think that's biological relevance? And since the main RNA molecule, they're not fully complementary, and then maybe in theories by looking at the coding, or maybe you in a like non physiological condition, they can interact, but inside the cell, maybe there's not that many interaction. Many of them is not maybe it's just theoretic. It's not happening inside the cell. What's your
So basically, for this interactome analysis, we overexpress mid-7D labeled with biotin for the pull down. When we overexpress something, that's normally much beyond than its biological presence. And this leads to more targets than in reality and a given specific time frame. And because of that, it becomes really essential that one should also validate some of the find targets by one-to-one gene analysis to really confirm. And of course, we started with 10 to 15 downstream potential targets, and we came up with 5, 6. Those were very consistent throughout our experiments, indicating that yes, the mechanism exists, but the question about all the interacting is the real interactor in a given specific time frame or not. This, of course, each technology has its own limitation, and we have to live with that.
okay? Okay.
Yeah.
Yeah, it's good to at the end you was able to identify the valid the pathways of polymers through the extensive study.
Because of that, it's really important to do the validation of your finding by different method and technology to really confirm that what you see is reproducible also in different experimental setup.
Okay. Yeah. Thank you very much, Dr. Singh, for such an insightful presentation today. And thank you for all the audience members for attending these webinars. And there are more exciting American Express webinars coming up soon. Our next confirmed speaker is Dr. Xu Wen Chao is from Taipei Medical University. He will be talking about the calcium phosphate nanoparticles for nucleic acids and the protein deliverings in primary culture and stem cell. So please stay into. So at MiCEE, we're committed to fostering a scientific communications and accelerating the pace of scientific discoveries. We thank you again for joining us today, and stay connected. And I look forward to seeing you all very soon. Goodbye for now. Thank
Yes,
you.
thank you. Thank you, Lian.
Yeah.
And just I wanted to say to the audience, if someone has a specific question, therefore feel free to approach me, and I would be happy to answer the question, whatever they have. Yeah.
Okay, thanks. Very kind. Okay. So, have a nice day. Bye.
Bye-bye.